Now I would like to introduce our first speaker. He is Dr. P.B. Bara Prasad. He is a university distinguished professor in crop ecophysiology, R.O. Cruz Indoor Professor in Agriculture, and Director of the Sustainable Intensification Innovation Lab at Kansas State University. He obtained his bachelor's and master's degree from Andhra Pradesh Agriculture University in India and PhD from University of Reading in the United Kingdom. He obtained his postdoctoral training at University of Florida. His research focuses on understanding responses of crops to changing environments, developing best management strategies to improve yields, providing food and nutritional security to smallholder farmers, and improving livelihood of people. Dr. Prasad is passionate about research, education, outreach, and enhancing human and institutional capacity around the world. He has active programs in multiple countries in Asia and Africa. He has published over 320 peer-reviewed journal articles and book chapters, trained 160 students and research scholars from 25 countries, and has given more than 100 invited talks in 40 different countries, including one of our first symposium here at the University of Florida in 2017 that you can find on our YouTube channel. His research has been cited more than 17,600 times. He received more than $150 million US dollars in grant funding and donations. He is an elected fellow of American Society of Agronomy, Crop Science Society of America, and American Association for the Advancement of Science. He serves on the International Commission on Sustainable Agriculture Intensification, and he is the past president of the Crop Science Society of America. Today, he will present on the title, Impact of Climate Change Factors on Crops and Opportunities to Enhance Resilience of Agri-Food Systems. We'll now share the floor with Dr. Prasad.
Are you able to see my slides?
Yeah.
Great, thank you. Thank you, Sudeep Kanwar, the Vice President of the Plant Science Council, for your kind introduction. Greetings, everyone. Special thanks are due to Josh Anderson, President of the Plant Science Council at the University of Florida, for inviting me and giving me this opportunity to speak to you today at this special symposium organized by the students and led by the students. Congratulations to him and his team for putting this together. It's a fantastic program. This is truly an outstanding endeavor and shows your leadership abilities. I also wanted to thank Corteva Agri-Science for funding these symposiums around the world and giving this great opportunity for the students. It was nice to virtually see and listen to Dr. Scott Angle, the Vice President of IFAS at University of Florida this morning. And I had the privilege of interacting with him in his previous roles at USDA NIFA Director and also the leadership of IFDC International Fertilizer Development Center. It was under his leadership at IFDC, the Soils Consortium was formed, and we were very glad to collaborate with and participate in it. And also congratulations, Dr. Angle, for being elected as a AAAS Fellow of the American Association for the Advancement of Sciences for your scholarly activities, leadership, and service. Well deserved. Some of you, as Sudeep mentioned, I did my postdoctoral training at University of Florida in the Department of Agronomy from 2000 to 2005 with Dr. Ken Booty and Dr. Hartwell Allen and Dr. Jim Jones. Most of them have retired now. But I'm going to show you 3 slides which summarizes my work, which I did there. And I wish I was there in person and learned from all of you. However, considering the conditions with the surge of Omicron variant of the COVID-19, I'm happy to participate virtually with you today. Considering the theme of the symposium and looking at what other speakers would be covering, I thought I would focus on climate change and its impact on grain crops and opportunities to enhance resilience of our agri-food systems. I'll keep my talk broad so that it'll be valuable to wider disciplines of students and young scholars who are thinking of advancing their careers in agricultural sciences. However, I'll cover some topics in depth. Considering the short duration which we have today, I'll only cover two components of the climate change, the temperature aspects and the carbon dioxide and their interactions, and show you some examples there. And of course, you all know there are other factors such as the drought, the UVB radiation, the ozone pollution, the salinity, which also impacts climate change, or climate change influences those. I won't be covering those today. So this will be my outline. First, I'll present to you the current status of global food security, malnutrition, stunting, and the crop yield gains which are happening now. And then we'll talk about the impact of climate change and climate variability, what are the past trends and future trends as well. And then I'll go into the details about how short time episodes of high temperature stress affects crops, and then what are the sensitive stages. And then we'll look into a longer duration stress and thresholds for different crops. And I'll explain the mechanisms of reproductive failure and lower yield in the high temperature stress. And then we'll discuss the interactions of CO2 and temperatures as well. And then I'll end by introducing you to the sustainable agriculture intensification concept, the meaning and its components, and briefly highlight, if time permits, other concepts such as broader One Health and using the principles of circularity in our agri-food systems. And these approaches can provide a holistic solution to address food, nutrition, water, energy, and climate security, and then end with some concluding remarks. Dr. Angle already got us started extremely well with his introductory comments about the need for food security, need for nutritional security, and also need for importance of gender and nutrients, and the role of US and also especially the southeastern part of US for food security. So let me start with the current status and issues related to food security, stunting, and yield gains. This is a world hunger map. At the current levels of population, globally around 811 million people are malnourished and do not have enough to eat. And most of these people are in Africa, South Asia, and Southeast Asia. In many countries in sub-Saharan Africa, about 15 to 30% of the population are food insecure. And this is not considering the impact of COVID-19 and other associated stresses caused by climate change. That may put another 20 to 30 million people under the poverty line. In addition to food security, we are facing serious issues with nutrition, particularly micronutrient deficiencies. For example, iron deficiency, which causes anemia, is widespread. And about more than 30% of the children in most under the age of 5 in most parts of Africa and South Asia suffer from this deficiency. In addition to iron, there are other nutrient deficiencies such as zinc deficiency and vitamin A deficiency, which are of significant concern. And due to those undernourishment and the nutrient deficiencies, we have serious problems of stunting in the children, especially young children. Globally, about 144 million children under the age of 5 are stunted, which is about 21.3%, which is unacceptable. And the three regions most impacted are South Asia, Eastern and Southern Africa, and West and Middle Africa, where more than 30% of the children under the age of 5 are stunted. And this particular visual is very important for all the agricultural scientists, and particularly to the plant breeding community. Although we have made huge advances in the yield gains of major cereal crops over several decades, especially in the mid and the later part of the last century, the yield gains after that period have been relatively slow and stagnated for most of the crops. Although we are seeing slight increases in the recent years, but to meet the demand of growing population by 2050, we really need to double the rate of gains of yield for major cereal crops. And also, let's not forget the legumes and the pulses and the oilseed crops as well. And we need to do this under the threat of climate change, which is intensifying both abiotic and biotic stresses. So that's a challenge for all of you. I'm sure you can do it. That takes me into the topic of climate change and climate variability. Climate change refers to the longer term changes in the climate, whereas climate variability refers to year variability or within season variability or occurrences of extreme events, which are much more damaging to the crop production systems. Over the last decades, there has been significant and rapid increases in various greenhouse gas emissions. And at present, the carbon dioxide concentrations are close to 415 ppm. Similar increases in trends have observed in other greenhouse gases such as methane, nitrous oxide, sulfur hexafluorides. These are extremely potent greenhouse gases, which even at a small quantities will have serious impacts on our current and future climates. Almost all parts around the Earth have seen increases in the global surface temperatures in the last 50 years. Particularly parts of the northern hemisphere have seen about 1 to 2 degrees Celsius temperature increases. If you look at globally, the global average surface temperature in 2020 were about 1.2 degrees Celsius warmer compared to the beginning of the last century. This may seem smaller, but this is a global average. In some parts, it was probably even higher, as I mentioned to you, especially in the northern hemisphere. Now let's look at the extreme events of the climate variability. Last 2 years, we saw extreme weather events throughout the world. And I just explain or give you some examples from US. The summer of 2021, especially the time period between June and August, temperature was the hottest in the US history, tied with the Dust Bowls. Similarly, in the year 2021, the percentage of land experiencing the drought, and especially in the western parts of the US, was extremely high. These extreme dry and hot conditions led to many wildfires, which devastated the natural resources in western part of the country. It also dried up many reservoirs around the country as well. And the year before, the 2020, we all know US had the largest number of named storms or hurricanes in the history. The number was set at a record of 30. And just looking at last month, in December 2021, you're all aware of the tornado outbreak where more than 60 tornadoes formed at the same time, covering this swath of about 200 miles in 9 states, which was unheard of. And we in Kansas, in December, recorded the highest December temperatures ever. So clearly climate is changing and is becoming variable, and we are seeing greater number of extreme events, which will be damaging to our food systems. How does the future look like? Most of the models predict that in the new climates, this will continue to happen, and we'll not only see the increases in the mean temperatures, but also see more occurrences of extreme hot or extremely hot weather over short periods of time. These extreme events are much more damaging to crop production if they occur at sensitive stages of crop growth and development. And when you look at the projections, by end of this century, if we continue with business as usual, most of the regions around the world will see increases in the mean temperature. Similarly, with the projected precipitation, in some parts of the world, you may see more precipitation, and some parts might see less. But in general, there will be more precipitation. But the evapotranspiration losses will be higher because of the higher air temperatures. So ultimately, the water availability will be lower, and this will cause drought stress to our agri-food systems. Now, let me move on to, as I said, that there's increases in the short episodes of high temperature stress, anywhere from few days to few hours. And we are already seeing that. And how does this impact the crop growth and development? Before I go into the details, I just wanted to bring everybody up to date about what does growth and development mean. As you know, growth is increases in the plant height or weight over time, whereas development is a change in stages during the crop period, from early to mid to late vegetative stages and the reproductive stages. I'm showing these stages for grain sorghum in this example, and emergence through flowering through physiological maturity. Not all stages are equally sensitive to environmental stresses. Some are more and some are less. And I conducted a lot of experiments and then we did a lot of research to identify the most sensitive stage. We know reproductive stages of crop development are generally more sensitive, but we wanted to know, even in that reproductive phase, exactly pinpoint which is the most sensitive stage. And to determine this, we did a lot of experiments where we exposed the plant to short periods of high temperature stress, anywhere from 5 days to One day, I'm showing you example of 5 days for sorghum here and the 1 day for peanut. And I'm just showing you these two crops, but we have comprehensive data on other crops as well. This visual x-axis is the timing of time relative to anthesis, 0 being anthesis, and the negative numbers are before anthesis and the positive numbers are after anthesis. And I'm showing you the data on the floret fertility as a percentage of control, 100 being the control under optimal temperature. As you can clearly see that when we expose the plants, sorghum plants, to 5 days of heat stress, it decreases the floret fertility at particularly 2 stages, at the time of anthesis and about 5 to 10 days before anthesis, where you see the maximum reduction in floret fertility. And for peanut, it's 1 day of high temperature stress. The maximum reduction occurs at anthesis and about 4 days before anthesis. These timings before anthesis coincide with sporogenesis, the microsporogenesis, the megasporogenesis, when the viability of the pollen grains and the ovules is being determined. And the time of anthesis coincide with the dehiscence, the pollination, the pollen tube growth, and the fertilization. And we have even shorter periods for the rice, where 6 hours of exposure at the flowering was significant enough to decrease the floret fertility. And more importantly, there was genetic variability among genotypes, and some were susceptible and some were tolerant, and some were actually escaping the stress by flowering very early in the morning before the heat of the day comes. So we need to explore that escape and the tolerant mechanisms. And those most sensitive stages for drought stress during the reproductive stages was also similar to that of temperature stress. Here I'm showing you the data on sorghum. Again, drought at gametogenesis and flowering decrease the grain numbers, whereas drought as early grain filling period decreases the grain weight because of the shorter grain filling duration. In combination of those lower grain numbers and the grain yield grain number and the grain size decreases the grain yield. So for most of the food crops, the sensitive stages are similar, first being a few days before anthesis at the time of sporogenesis and pollination and fertilization. And those stresses, the other stresses like cold stress and salinity stress also have similar sensitive stages. So now let's move on to what are the mechanisms associated with this reproductive failure in most of the grain crops. And I'm going to show you broadly what the mechanisms are. But I understand that there is a talk by Dr. Kevin Bexey from Plant Biochemistry and Physiology Department at the University of Florida, and he'll talk more about the mechanisms in detail. So I'll just provide you a broad overview. If you look at the mechanisms of reproductive failure, there's five various ways in the viability or the seed numbers are decreased either due to loss of floret fertility or poor fertilization. The first is that at high temperature, the reactive oxygen species are decreased. The reactive oxygen species is increased, and then the antioxidant ability is decreased, leading to the membrane damage, and especially for the pollen and the ovules. And then that leads to the sterility, and then leads to loss of seed set, decreased seed numbers, and finally decreases the yield. The second is at high temperature stress, the lipids in the membranes, particularly the oxylipids, are increased. And then the membranes are damaged and the loss of viability occurs. And the third is the sugar metabolism. And especially under high temperature stress, there's the downregulation of hexose transporters, which actually moves the sugars from the anther wall into the pollen grains. That starves the pollen grain and leads to the sterility. And the fourth is related to the pollen and stigma anatomy, where the exene wall of the pollen is changed. And then and also there is a rapid degeneration of the tapetal cells, which provides the nutrition to the pollen grain. Once that is disintegrated, the functionality of the pollen decreases. And the fifth is related to the reproductive morphology. Under stress, what happens is the dehiscence of anthers is affected. And sometimes we see stigma exertion, meaning that the height of the stigma is higher than the anthers. So there is not enough pollen reception by the stigma is impacted. And we see that in many crops. And that leads to lower seed set. Now, quickly looking into what is the longer duration impact of these high temperature stress and thresholds for various crops. Different crop species have different optimum and ceiling temperatures. and have different response functions as well. I'm here showing you some of the curves for major cereal and legume crops. I chose to represent harvest index because it captures all the components and what is happening throughout the season. We can see crops vary in their optimum and the ceiling. Optimum meaning where you have the highest grain yield or the highest harvest index. Ceiling meaning that where there is no grain produced and the harvest index is 0. So among cereals, wheat is the most sensitive with a ceiling temperature of about 31 degrees Celsius, followed by rice at 35, sorghum at 38, and pearl millet at about 41 degrees Celsius. So pearl millet is one of the most heat tolerant cereals. Similarly, among summer legumes, dry bean was the most sensitive at 32 Celsius, and peanut at 39, and soybean at 40. And I've not shown you the data on mung bean and cowpea, which are actually even more tolerant and has a ceiling temperature of about 42 degrees Celsius. So with climate change, we need to be changing and adjust our crop species selection based on the environmental conditions. And here are some of the mechanisms through which the long duration high temperature stress decreases the grain yield and the harvest index. High temperature stress generally causes increase in the canopy temperature, and it also causes the higher vapor pressure deficits, and these leads to decreased photosynthesis and increased respiration. And it also causes early leaf senescence and also shortens the season, which leads to decreased biomass production, lower light interception. And as I already told you, that high temperature causes a reproductive failure, leading to lower grain numbers. And high temperatures also decreases the grain filling duration, resulting in smaller grain size. Those smaller grain size and the lower grain numbers ultimately decrease the grain yield and decreases the harvest index. So that was the temperature impact. So now let me quickly move on to the carbon dioxide and the interactions between carbon dioxide and temperature, because that's what you're likely to see. So I'll show you two crops. One is a C4 crop, C3 crop, peanut, and another one will be a C4 sorghum. First, let's look at the photosynthesis, and then later we'll go on to the other traits. On your left is a response of groundnut leaf photosynthesis to elevated carbon dioxide at different air temperatures. The pattern for the next few slides will be the same, meaning that x-axis has a temperature, and then the y-axis has a trait. And then you have two lines representing carbon dioxide levels, blue being ambient and then red being the elevated. So for groundnut or peanut, under elevated carbon dioxide, the leaf photosynthesis increases across all the temperature. And it was substantial, about 20 to 30% increase. And there was no negative effect of temperatures on photosynthesis. While on this grain sorghum on your right, the C4 crop, you have two panels. The top panel is at optimum temperature 30 by 20 degrees Celsius. And the bottom one is a 36 by 26, which is day and night temperatures. And then the x-axis shows you the age of the leaf or the days after sowing. At early stages of leaf development, carbon dioxide slightly increased the leaf photosynthesis, but at latter stages, there were no response at all. And you know the reasons for that, because sorghum being a C4 plant and the presence of trans anatomy and the efficiency of PEP carboxylase, you don't see generally response to elevated carbon dioxide. So another example here, now I move on to the other traits beyond just photosynthesis. Here I'm showing you the data on common bean. For this, increasing temperature decreased the total dry weight, the seed numbers, seed size, and the seed yield. But there was interactions between between temperature and carbon dioxide. For total dry weight, elevated carbon dioxide increased the dry weight across all the temperatures. So you see those parallel lines. While seed yield, the benefits of CO2 was observed only at the cooler temperatures and not so much at the higher temperatures. So you see the converging lines. And for seed numbers and seed size, there was no effect of elevated carbon dioxide at any temperatures. And this is a response for the C4 crop, grain sorghum. Again, increasing temperatures decreased seed set and the seed yield at both ambient and elevated carbon dioxide. But in this case, more importantly, elevated carbon dioxide decreased the seed set even under higher carbon dioxide across all the temperatures. This is primarily due to the increased tissue temperatures caused due to closure of stomata. And similarly for other crops, elevated carbon dioxide, similar to the other crops, the elevated carbon dioxide increases the seed yield at a cooler temperature, but not at higher temperatures. And when you look at the sensitivity of the seed set the ceiling temperature, that's when you don't have the seed setter. The ceiling temperatures under elevated carbon dioxide is about 2 degrees Celsius lower, as represented here for common bean on the left and then the rice on the right. This suggests that elevated carbon dioxide is not good for reproductive function. So most of this research was done at the University of Florida in the agronomy department. So this is why I just wanted to show you some of those items. So now let's quickly move into the climate change and what is the impact of climate changes on the biotic stresses, especially pests and diseases. And I'm just going to give you one example for each of these, one for insects and then one for diseases. For insect pests, I would like to give you an example of infestation of desert locust in 2020, just a few years ago. The outbreak was one of the worst in the Eastern Africa, probably the biggest in Kenya in the last 70 years, and even in India in the last 25 years. And this was caused by unusual weather conditions the previous 2 years before 2020. That was extremely wet with a lot of cyclones in 2018 and 2019, and the irregular sea surface temperatures in the Indian Ocean because of the Indian Ocean dipole, which is primarily caused by the climate change. The outbreak of this happened, and this caused huge losses to crop production in many countries in Africa and Southeast Asia. So that was for and there are other examples of rise of blast or rust and many other diseases as well. So, but I'm going to, for a disease perspective, I'm going to show you an example of coffee rust in Latin America. This disease generally spreads at a temperature in the range of 18 to 23 degrees Celsius. And these conditions generally existed in the Latin America region in lower altitudes in the mid-1990s. So it was limited to those regions. But due to climate change, even the higher altitudes started to see these increased temperatures. So therefore, the disease spread in that areas and decreased the yield of coffee, both in terms of quantity and also the quality of the coffee. And again, you all know the disease triangle, the susceptibility, the host, and then the environment. And those dynamics influences, and those are being changed with the climatic conditions. So those were the effects. And then now I will talk a little bit about how do you manage this, and then just highlight some of the breeding approaches. We can manage these abiotic and biotic stresses by both agronomic tools and the breeding approaches. First, I'm going to give you a broad visual. This illustrates a lot of things, but the strategies range from agronomy to crop management. That includes appropriate time of planting, optimum plant population, efficient use of light, appropriate irrigation management practices, balanced nutrition for better plant health, and managing pests and diseases. And we can also use crop rotations and crop mixtures to manage pests, disease, and weeds by breaking their cycle. Then we have also options with chemicals and use of microbials, such as growth hormones, growth regulators, antioxidants, osmolytes, and now nanoparticles, and microbial area is significantly increasing, especially to manage the abiotic stresses, how plant adapt to. And then they enhance the plant's ability to tolerate these stresses. And we also have options of variety selections in terms of those varieties which escape a shorter duration cultivar, or early flowering, or early morning flowering to escape the hot temperature stress of the mid-afternoons. And then cultivars have some cultivars have shown tolerance in terms of whether their pollen viability or the pollen tube germination occurs at higher temperatures as well. And of course, we all know about the greater photosynthesis and longer green leaf area duration, and also the ability to maintain carbohydrate production and partitioning to the grain. And there's also genetic variability for roots for water absorption, and mainly cooling the canopies and improving the harvest index. So largely, with the use of tolerant genotypes, I've listed many traits here, such as gamete viability, spikelet fertility, which is similar, rapid grain filling, rates, increased seed size, canopy temperature depression, and also the stay green traits. So these are some of the options which you'll find in the literature, and you can find more details there. And from the breeding perspective, there's been a lot of improvements which are happening. I'm not an expert on this, but I think I'm sure many of you participating in this understand this better. And I know that Dr. Jeff Morris, professor from Colorado State University, is one of the speakers. He's an outstanding researcher. I'm sure he'll provide much more details into this stress adaptation. Similarly, I believe that you have a talk from Dr. David Bubeck, research director of plant breeding and at Corteva, an excellent scholar again. They'll provide you more details about the targeted breeding. I'm just going to give you a broad picture and a broad summary. So focusing on exploring or screening large genetic resources to identify tolerance is going to be key steps. And we need to explore both close and the wild relatives to identify tolerance and then use them in the breeding program. And the recent methods of fast breeding, speed breeding, or double haploids is shortening the time for breeding, which will allow us to produce more rapid generations. And again, we can use both traditional breeding and the new approaches. From a molecular breeding perspective, the genomes of most of the important food grain crops have been sequenced, and we can now efficiently use the genomic tools, the genome-wide association mapping, whole genome sequencing, to identify some of the tolerant traits and the genotypes thereafter, and improve the efficiency of the breeding methods. There's also transgenic approaches with the introduction of new genes from different crop species, and also using genetic transformation technologies. And these are gaining importance. And you're all aware of the new gene editing approaches with CRISPR-Cas9 technologies, which adds another tool to our toolbox. And there's been significant progress made in some of the crops for improving abiotic stress tolerance using those CRISPR-Cas technologies. And again, I'm sure there are other speakers who will talk more eloquently about these topics, and you will learn from them. So before I have, let me check the time. Okay, so let me now give you a systems approach. Let's move beyond mechanisms, impacts, and breeding to how to actually address and use systems approaches, because we have complex problems with food security, nutritional security, climate security, food waste, and then we need novel platforms which can bring all of us together. This is because of the complexity of the agri-food systems, from production to consumption. So I will introduce and put forward to you three approaches if time permits. I'm at 30-minute mark, so maybe 15 minutes I should be okay. So I will introduce you three approaches. First, I will start with a broader One Health concept, and then I'll introduce you the concept of circularity or circular economies, which is gaining significant interest. And then the last one will be the one which I am more focused on, which is sustainable agricultural intensification. So the first one to consider is a broader One Health platform. And all of you, I know so many of you are members of the Tri-Societies, the Crop Science Society, American Society for Agronomy, and the Soil Science Society of America. Recently, all three societies came together about 2 years ago, and then we developed our common grand challenge statement, which reads as drive soil, plant, water, environment system solution for a healthy planet, for healthy people on a healthy planet in a rapidly changing climate. So to address this, we must work all together and then identify some frameworks which can bring us together. So using a broader One Health platform can bring us together. At present, the broad One Health includes only humans and animals and ecosystem. But we all know for better human and animal health, it all begins with healthy soils, which leads to healthy plants. And all together, healthy soils, healthy plants, healthy animals, and healthy humans will ultimately lead to healthy ecosystems. And the circularity continues. And all of these are interconnected, both in terms of positives and also in terms of negatives. In this visuals, I've shown you some positives in the green color, and then few negatives in the red. The positives being the nutrient cycling, the yield, the nutrition, the health, the species diversity, and the ecosystem services, which each of these provides, and the negative being the pollution, the toxicity, the pests and diseases, issues with food safety, the degradation of land, natural resources, and the loss of biodiversity. So those are the negatives. And then COVID-19 also reiterated the need for broadening this One Health concept as we prepare ourselves for any future disease or outbreaks from different species. COVID-19 was one one virus which jumped from animal species to human species. So there are several others which can spread between multiple species in the biomes, right from microbes in the soils to plants to animals to human beings. So we need to be really taking this systems approach and think broadly just beyond one or two species. And the second platform is the circularity, or the focus on system redesign. Because in our system, increasing input, the focus should be on increasing the input and the resource use efficiency throughout the system from production to consumption. And we know that in agri-food systems, there are large inefficiencies, either due to loss of inputs or poor choices of inputs. And of course, there's a lot of issues with food waste and other resource waste as well. So we have nutrient losses, we have low nutrient use efficiency, water use efficiency. So we have lots of opportunities to improve the efficiency by redesigning the system and using the principles of circularity. And also, we have options of using the principles of 5 R's for water and nutrients, using the right source at a right rate, at a right place, at a right time, in the right manner. That applies for both water and nutrients. In addition, we can also eliminate losses in the waste through recycling, reuse, repurposing, and residue management as well. I do not have time to show you some specific examples, but this circular economy is very relevant to agricultural sciences, and it can be applied to the carbon cycle, the water cycle, the nitrogen cycle, the methane cycles as well. So, and I believe that there'll be other speakers who can probably talk to you about this one. The third and the final one is the concept of sustainable agricultural intensification, which actually captures many of the concepts which are already, as I mentioned, in the One Health and the circularity. The goal of sustainable ag intensification is to provide access to safe, nutritious, and healthy food at all times to all people from existing farmland without damaging our natural resources and ecosystem health. So the bottom line, main point here is existing farmland and actually not damaging. And I'll go even one step ahead, but actually go ahead and make a positive environmental natural resources, which regenerative agriculture, which is happening. So again, it has many components. It can broadly be divided into three boxes. Here I'm showing you in the genetic intensification, which is in the red, that is focused on increasing the yield, building tolerance to abiotic and biotic stresses, improving nutrition by using both traditional breeding and novel genomic and gene editing tools. And then the second box is agro-ecological intensification, which is in the green, which is more focused on diversification, efficient use of crops, efficient use of farming systems, efficient cropping systems, improved agronomy, the soil, the water, the nutrients, and improves resource use efficiency, and also taking a more integrated approach for nutrient, soil, water, and pest management strategies. The third being the socio-economic intensification, which is in the blue, is more focused on developing markets, policy interventions, understanding the barriers of adoption, building the social capital, creating enabling environment, and institutional building. And all of these are equally important. And then there are other innovations which cut across all of these three, like the digital tools, the geospatial tools, the artificial intelligence, mechanization, precision agriculture, and entrepreneurship, private sector engagement. So it's all very important. And basically what this whole concept covers is captures the interaction between the genotype, the environment, the management, and the social components. And of course, there's the use of big data, the communication, the youth, and the gender is an integral part of this. So SAI provides us a holistic platform to address issues related to climate security, food security, resilience, and climate action. So that's one of the last systems things which I wanted to share with you. My Sustainable Intensification Innovation Lab is focused on many of those components, and we are funded by the Feed the Future initiative of USAID, the U.S. Agency for International Development. It is one of the largest innovation lab, and we have a very broad portfolio, and we take systems approaches, some of those three which I mentioned, and we have active research, education, and outreach programs in multiple countries in Africa and Asia. I don't have time to go through this today, but I'll encourage you to visit our website and learn more about our activities. Let's see the time, 37 minutes. So maybe if I will take about 3 more minutes before I wrap up. So I think I've showed you the challenges, and I've showed you some approaches for understanding the stress tolerance and the breeding approaches and the systems approaches to bring us all together to address the multiple issues which we're talking. So the big question is, what are we doing? Are we doing enough? Particularly in the global south, where we have major challenges of food security, nutrition security, and those will be further impacted by the climate change. So I would like to bring to your attention, there was an International Commission on Sustainable Ag Intensification, which was established in 2020. It was for 2 years. So we are wrapping it up right now. And I was honored to be part of this commission. The goal of this commission was to identify and recommend priority areas to strengthen agricultural systems and to meet the sustainable development goals and the climate needs. And then again, as Dr. Randall mentioned, that technology is the key to solve these issues. So we wanted to know, what is the current status of spending in innovations? How much are we spending on innovations? Because unless you have those resources available and are invested to come up with the technologies, it will be difficult, right? And then also we want to look at how much of those spends are in the biophysical, the social, the human aspects. And again, they have produced several reports, and you can please visit the website. And then there are several aspects both from the biophysical and the social science perspective. But and they are freely available, and you can download them easily in PDF forms as well. There are also some policy briefs there. But I'll just, in the next 2 minutes, I'll just highlight you two main questions, or at least 2 answers to 2 of the main questions. The first question was, what is the current status of investments in innovation in agriculture in global south, meaning that the less than the middle income countries? And who are funding these initiatives? Dalberg Asia produced this report, and I chaired the oversight group. So our results show that about $60 billion is being invested annually on innovation, technology, innovation, right, in agriculture in the global south. But only 7% of that aligns with environmental outcomes. And only half of that, meaning 3.5%, aligns with social and human goals. This is extremely low and needs to be changed if we want to meet the Sustainable Development Goals by 2030. And the greenhouse gas emission goals of the Paris Agreement will limit the warming under 2, or actually under 1.5 degrees Celsius. There's a lot more details in that report, so please visit the website and look at those details. So that was about what is innovation funding currently happening in the global south. The second question is, what is the investment gap if we were to meet some of those SDGs to remove hunger and to remove climate goals? And of course, there's many reports which have been developed in the past. And this report was developed by Dr. Mark Rosegrant and his team at IFPRI, International Food Policy Research Institute. There are several numbers floating around, but this is most conservative numbers, and we can actually make some changes here. So they found that the gap is about $15 billion a year. If you're able to invest and redirect this amount yearly up to 2030, we can minimize the hunger below 5% in most of the regions, with an exception of sub-Saharan Africa, where we can bring it down from 30% to 11%. Not only that, we can actually significantly decrease the greenhouse gas emissions, the agriculture water use, and the deforestation. Those numbers are on these visuals. And again, please visit the website, and it will have a lot more details. So clearly, a modest investment of $15 billion focused on research and scaling, we can remove hunger and address many of the climate goals. So now I'll conclude. Sorry, one more. So hopefully I've convinced you that climate change and climate variability is happening. We're all experiencing in our daily lives, and it will have negative impacts on grain yield of major food grain crops in most of the regions, and especially the short episodes of abiotic stresses, if occur at critical stages of crop development, will decrease yields and cause significant yield losses. And food grain crops are particularly sensitive at 2 critical stages of development. One is happening a few days before anthesis. That's when the gametogenesis, the viability of the pollen and the gametes is being determined. Second being at the time of flowering. That's when the pollination, the pollen germination, pollen tube growth, and fertilizations are happening. And then the third, for some, is the early part of the grain filling, where the rapid grain filling starts. And if you have stress at that time, it decreases the grain filling duration as well. So the yield decrease under these primarily occurs due to the lower decreased grain numbers and the grain size, and sometimes to a little extent to the lower biomass production if it is a season-long temperatures. And then the shorter grain leaf area duration because of early senescence, and also lower partitioning to the grain. And we saw that there will be benefits of elevated carbon dioxide for photosynthesis and the biomass, vegetative biomass, but those benefits will be negated by high temperature stress. So there will not be any benefits of elevated carbon dioxide, especially they're accompanied with higher temperatures. You may have the benefits of the cooler temperatures, but not at above optimum temperatures. And then there is genetic variability, and but the parental lines which are currently being used is limited. So we need to systematically explore the germplasm collections, including both the wild species for tolerance, and use them in the breeding programs. And of course, we all know that we need to have high throughput phenotypic tools and molecular and identify molecular or biochemical markers so that we can actually screen large germplasm collections. And the beauty is that now you have the genome sequence, and then we need to match the genomic data with the phenotyping data as well. And more importantly, the interactions of multiple climate change factors, the temperature, the water, the radiation, the vapor pressure deficit, and the photoperiods needs to be understood more clearly, because all of these are going to happen together. And not only that, it also have these also will impact the biotic factors, as I gave you examples of pests and diseases, and how does those two interact with each other, with the host susceptibility, the environment, and the prevalence and intensity. So those needs to be understood well as well. And the last but not the least is that climate change will not only decrease the yield, but also it will have negative impacts on the grain nutritional quality. So we need to better understand how those climate change factors will actually impact nutritional quality of the grain that will eventually affect the food nutritional security of the people. And then we need to combine the breeding not only to improve the yields, stress tolerance, but also looking from the perspective of grain composition as well. And finally, transdisciplinary research using systems approaches such as the SAI, the sustainable ag intensification, the broader One Health, and the circularity must be used. And the collaboration between crop scientists, agronomists, and soil scientists will be critical. Naturally, we need to go beyond biophysical sciences and work with social sciences to ensure that our new technologies are scaled at a level where we can have an impact on food and nutritional security. With that, I will end. And if you would like to learn more about our activities, please visit our website and follow us on our social media. And again, I'll be glad to take any questions. We are at 45 minute mark. Thank you so much again for this opportunity.
Thank you, Dr. Prasad, for the excellent talk. It was such a pleasure to hear from you, and I'm sure that all of our participants feel the same. We have few questions here in Q&A, but we are running a little bit out of time now, so we'll go just for our questions. I can see here one question that, are there any high throughput phenotyping platforms that you are aware of and excited about in terms of their ability to tackle issues of maintaining sustainable agricultural production system in the face of climate change?
Yeah, I mean, that's a great question. I think we all have a very good genomic tools, but I think you're talking about the high throughput phenotyping tools. With the UAVs and the drone technologies, now we are able to put cameras, high resolution cameras with digital cameras as well, and also the other sensors where we can have a spectral imaging, where we can take images and look at individual plots, not only individual plots, but actually individual plants, to look at the green leaf area, the greenness of it, that is directly related to the senescence. And also thermal imaging will tell us how cool the canopy temperatures are. So I think there are opportunities to really use high throughput phenotyping techniques with UAVs and also satellite imaging in some cases.
Thank you very
Mostly
much.
from the perspective of the plant health.
Okay, thank you very much, Dr. Prasad. Since we're running out of time, could you please go through other questions and can type some answers like in short? Yeah, thank you very much. I appreciate that.
Yes, let me respond to one question. Can sustainability and enhanced production go hand in hand? Yes, absolutely. That's the concept of sustainable ag intensification. The fact that both are together, that means we can have increased production, but also take care of our environment under the face of climate change. Thank you.
Okay. Thank you very much. I appreciate your awesome talk.